Good afternoon. It's my pleasure to introduce Rodrigo Weber Dos Santos from Brazil, who will tell us today about modeling cardiac perfusion and myocardial edema using formulation for porous media. Rodrigo, it's your word. Thank you very much for accepting our invitation.
Thank you, Julia. So it's a pleasure. Thanks a lot for the invitation. It's a pleasure to give this talk. Unfortunately, it's not on local. It's remote, but I was lucky enough to be there once, to be in the University of St. Petersburg. So I know it's a beautiful city and a very nice university. I hope to come back soon one day. So as you mentioned, I'll be talking about modeling cardiac phenomena in general using porous media. And as I read, so I'm from the Federal University of Juiz de Fora. Juiz de Fora is like a mid-sized city in Brazil. We have around 600,000 inhabitants. The altitude is like is around 1,000 meters. So it's not, we do have winter in Juiz de Fora, different from Rio de Janeiro that does not have a winter. We do have like 0 degree, 5 degree during the winter time because it's the high altitude. And this is Juiz de Fora. It's really in the middle of the biggest capital in Brazil, Rio de Janeiro, São Paulo, and Minas Gerais. So it's close to the biggest capitals in Brazil. So the Federal University of Juiz de Fora, this is the campus side. We have 1,000 professors and around 25,000 students with many undergraduate and graduate courses. And we also work in our, I have collaborations with, I had projects before with Petrobras, the oil company here in Brazil. We have a project now with Shell Brazil, together, so coordinated by Gregory Shapiro, that was also a former student from Dan Marquezin at IMPA, like myself, together with also some guys from PUC, Rio de Janeiro. And we're very active in these recently in foam-based enhanced oil recovery and uncertainty quantification, which I think is very near to, at least reading the description of your site, is near to what a lot of you are doing in St. Petersburg. But Julia asked me to present something we are doing with cardiac modeling, which is something like different. So I've been very active in modeling the heart in the last 20 years or so. And we have developed different models for different kind of phenomena in the heart. The heart is really amazing in terms of what you can try to model, because the contraction of the heart is actually always synchronized by the electrical activity. That's what I'm showing here, the electrical activity of the heart. And here also the electrical activity of the heart that then synchronize all the mechanical contraction of the heart. And together with these, we have also perfusion in the heart. We have inside the cavity, we have like fluids in the big cavities of the heart. So we have fluid mechanics. And then we have different diseases, and we do model like edema formation, so water accumulation during infection of the heart, to myocarditis. We model like infarct in the heart, bad perfusion, and I will try to focus on these two here: cardiac perfusion and edema formation, which are two applications that involve formulations of porous media. in the heart. But actually, a lot of these applications involve porous media formulation. Can be formulated as porous media. Like most of the biological tissue, yeah, any biological tissue, you can, and depending on what you're interested in, you can look at it as a porous media also. So here is like a post-COVID problem from a lady of 26-year-old, and you see here, this is called an exam called magnetic resonance imaging that you do with contrast, that you can identify edema, like inflammation in the heart. So you have like a lot of water accumulation in the heart, and this has a lot of impact in the how the heart works. It can be very bad for the patient. And so we're gonna focus on these kind of exams, magnetic resonance imaging, to see, to try to get these images, which are very complex to analyze. Here's the left ventricle of the heart, and here's the right ventricle of the heart. Just a short cut, 2D cut of these organ. And here it's the medical doctor just visually identify like marks. Oh, I see here edema. So it's really based on how good is the medical doctor to get to interpret the image and try to see. And we try to use models exactly to try to quantify a little bit more this process of a diagnosis of different disease in the heart. So myocardial infarction is another disease. So you have normally the heart also has to perfuse itself. So it pumps blood to all the bodies, to all the body, and to all the organs, and to itself also, with the main coronaries. Yeah. And when you have a block of the coronary, so can be due to genetics or just bad habits of not exercising or things like that. You can have these block in the artery, in the coronary, and these will, so there will be an impact. A lot of tissue, not a lot of tissue, but some tissue, part of the tissue of the heart will suffer, and some cells will die. When the heart is suffering, or the cells is suffering, you say ischemia. Ischemia means it's suffering, and the infarct, when it's an infarct, it's mean it's already dead. And then if you take histology slices in this part of the heart that is damaged, you're gonna see different topologies, depending on how was the infarction, and also depending on if it's not only infarction, it's for instance a virus or bacterial inflammation. You're going to see different patterns like this one, like colony of bacteria here damaging the tissue. And so the red is like normal cells here, and anything that's not red is white or blue, shouldn't be there. And so it's because the heart is being damaged here. So it's interesting that was like a big mystery, and still is, like, because when you actually, the part of the size of the damaged tissue is not big. So if you have a infarct, it's like 1% of the tissue of the heart is damaged only. But somehow, because of these 1%, you can get very serious complications of your health, and it can really die because of these 1% of tissue that is dead. So it's always been like a great challenge to the medical doctors And so in 73, there is these Boineau and Cox. They had these a lot of experimentations with animals, with dogs, and they postulated that these what makes the heart to stop because of even infarct, it's actually because of the electrical activity that is desynchronized. So it's not, so you have the electrical activity, like there's a very fast wave of electrical activity that says, oh, it's time to contract all the cells. So it's the trigger to synchronize and the contraction. So if this electrical activity is desynchronized, so the heart doesn't pump blood well anymore, and then without the heart pumping blood, after a few minutes you die. And these Boineau and Cox postulated that these happen because of these micro reentries in these damaged tissues. So there will be like zigzag propagations of the electrical wave that would break this the synchronous activity of the electrical wave in the heart. And this was postulated in 73, and still it's still a theory. There's no one knows exactly what happens during what's desynchronized the heart. There's many theories, at least 3 big theories that explains that. One is the micro reentry, and our group was the first group that managed to recreate these micro reentries almost 50 years later, after Cox in 73. We managed to simulate an infarct, and there's a propagation of the wave, and it breaks here. And there's very desynchronized activity inside this damaged tissue, and these will re-excite the heart here. It's leaving to re-excite the heart, and then it starts to re-excite and re-excite, so very fast, and then your heart is just shaking. So you have a shaky heart instead of a beating heart, because there's different sites of waves, electrical waves, and then you die. And this can be because of fibrosis, fibrosis means increase of the connectivity tissue. So there are heart cells are called myocardial cells, and they are connected by this extracellular matrix, which is called connective tissue also. And when the cells are dying, they're replaced. They're being they cannot be avoided there. Yeah, so they're being replaced by extracellular matrix. The increase of extracellular matrix is called fibrosis. And we have shown that fibrosis and ischemia, so fibrosis and lack of oxygen, can really can explain a lot of different diseases, even like a very common disease that can be very dangerous also, like sleep apnea. So if you have if you snore too much because you're not breathing well during your sleep, you're constantly in ischemia, make your heart okay, you don't have enough oxygen and your heart can be suffering. And these alone can trigger all these processes of very dangerous arrhythmias. And we were, like I mentioned, we were the first to reproduce these with a patient-specific heart. We brought a patient, magnetic resonance image from a patient that had an infarct, and we segmented the images to generate the mesh. We have performed all this process to generate a personalized or patient-specific mesh for that patient. And we could explain via this micro reentry mechanism why the heart completely leaves lose the synchronicity, and there's this turbulent wave. It's called fibrillation that can kill you. And that was published also in 2018. So yeah, but in general, the heart is heterogeneous anyway. So this heterogeneity can be bad, like I show, but in general, the heart is heterogeneous. There are a lot of different tissues that compose the heart. There are the vessels, there are the extracellular matrix, like I mentioned before. There are the myocytes, myocardial cells, and it's a patchy heart. And here's just some components of these patchy heart. So you have different colors here for the myocardial cells, the cells that contract. But then you have like the vessels here in red. You have the extracellular matrix in blue. It's a lot of extracellular matrix that brings all these components together. And you can have some other cells like fibroblasts and other cells also inside your heart. So of course, when you look at these and you see like, okay, I want to model perfusion, and the perfusion will be contained in the vessels. So you can look at these and say, okay, this is a porous-media. So there's the blood will be always inside the vessels, and there will be a lot of void space around, and you can consider it a porous-media. Or if you model like bacteria spreading here in the extracellular matrix and attacking the cells, so you can, okay, there's another porous-media. So depending on what you want to model, so you take these different patches of these, or different domains of these complex tissue, and can address the modeling of the phenomena using porous-media. So the goal number one here of this talk is just to reproduce something that's called perfusion using a contrast agent that is used both in the magnetic resonance images and also in computational tomography images for the heart. So this is a magnetic resonance imaging, and this is called the first perfusion. So you inject a contrast and the heart goes all bright. If you see something black, it's because this tissue is not being perfused. So the blood is not, the contrast is not arriving there, which is an indication that blood and nutrients and oxygen is also not arriving in this tissue. So it's bad. And there's another, if you wait like 30 minutes or so, 10, 20 minutes or so, and do again. And if you see that the region that was black before is now very bright, then you can be sure that this tissue is damaged, is dead, because the process here, I'm gonna explain in the next slide what's the process here with the contrast agent and the tissue. But clearly you see there's a lack of quantification. So it's very complex images that the medical doctors have to use to make a diagnosis based on these images. So our idea is really to provide models to help the clinical clinical and clinicians and doctors to perform diagnosis with more accuracy. So this is what's happening with the contrast agent. So if you have a little bit of contrast agent, this specific contrast agent, it goes outside of the capillaries of the arteries to the extracellular matrix or interstitial space. Also, I call it these interstitial space along the tunnel. And but this contrast does not invade the cardiac tissue, the cardiac cells. So here we have contrast agent leaving the capillaries and going to the interstitial space. And normally, if the heart is fine, it will go back after a while, go back to the venous system and then be washed away, washed out. What happens if there is replacement of, so this cell is dead and it's replaced by extracellular matrix? So what happens is that this contrast agent gets trapped in this extracellular matrix. So it cannot leave anymore. So that's why you do these 20 minutes after do the exam. If you see a lot of contrast, it's because there are no cells anymore in that part of the tissue. All the contrasts were trapped in extracellular matrix, so the cells are dead. If you do after 20 minutes again, and there's no, so if it's bright, it's because the tissue is dead. If this is not bright, it's because the tissue is still viable. So it means it's the tissue is suffering. So perfusion is bad, but the cells are not dead yet. So it's a very important exam. So that it's a decision. Then you make a decision. So okay, I'll go to catheter. I'll put a stent in the. I'm gonna put a stent here in the vessel that will expand the vessel so that the blood perfuse better, and I'm gonna save that tissue that is not dead yet. So, but if the tissue is dead already, so maybe it's not worth putting all the stent or doing a procedure because it's too late. So it's these kind of exams are very important on how I'm gonna handle the patient. And so the first model is we're going to try to reproduce these exam, all these phenomena here. So we're going to treat the heart as a porous media. Actually, there's these complex tree of the arteries. So the contrast agent goes inside, but also leaves this contrast, these tree of arteries and vessels, leaves to the interstitial tissue, which is this white part here. It can leave to this interstitial tissue, but they cannot enter these particular contrasts cannot enter the cell. So we have like a double porosity media, like we have two levels of porosity here. Yeah. And we're going to try to make, so this is the kind of simulations we're going to perform using our models. So here's so we have like many domain model we propose. So we have a domain for the arterial domain, porous media, you have a capillary domain with different porous media. And then there's the exchange of flux here. Then here in the capillary domain, there's exchange between the capillaries and the interstitial space, as I mentioned. Yeah. So there's this exchange between these, the capillaries and the white space here. That's the space between the cells, interstitial space. And if there is dead cells, there will be an infarction here. So there will be a lot of extracellular matrix that will trap also the contrast agent here. So, and then from here you go to the venous system, and then there will be a recirculation all over your body. So the contrast will be eliminated a little bit by the kidneys, but then we will enter again, and in the exam you see the contrast going out and back. So going to zero, it will be washed out of your system, but it enters the heart many times. So we also managed to capture these many oscillations also with our model, and we published these in two years ago. So this is like a simplification. Another paper first we simplified. We collapse all these domains here. We collapse into one domain just to simplify. And we have this domain here, which is the intravascular domain, communicated to the extravascular domain, to the interstitial space. And then there's the outflux to the whole body. And then there's this, this is like really like a one-dimensional model just to make this connection, like this recirculation between of the contrast. And yeah, that's the model I'm going to discuss now. So we have this intracellular domain, omega I. And we assume here that mainly there is the intracellular domain is a porous media because it's the vascular domain, the vessels. And we assume just a Darcy law. So the velocity is proportional to permeability divided by viscosity. Here, the K is not it's not permeability. Then it's a hydrostatic tensor times the pressure gradient. And here, so you have the contrast in the intercellular space. So this is the concentration of the intracellular, the constant in the intravascular, sorry, in the intravascular space. And here's just a mass balance for the constants inside the intravascular space. So it varies because it's being advected, it diffuses, and because it loses, it loses mass to the interstitial space. So there's an exchange of contrast between the intravascular space and interstitial space. In the interstitial space, you have 1 minus phi, so it's the other. So if this is the porosity that represents the vascular system, 1 minus phi times lambda is what's left as space for the contrast agent to leave, and in the interstitial space. And we neglect advection effects because it's known that advection is really mainly when you have inside the vessels. Outside the vessels, you have mainly diffusion only process. So you have diffusion process. You have, again, the same exchange here of mass between these two systems by this F. And we're modeling the venous system here just by capturing these constants here. If we have damaged tissue, we have another domain, which is the fibrosis domain. So it's the extracellular matrix domain. Then there will be an exchange between the interstitial space and the fibrotic space. Another exchange rate here, G, which is just linear for this simple model. And this is like just a very simple equation for adsorption. So just we model like there's no movement of the contrast. The contrast will be trapped by this in the extracellular matrix in the fibrotic domain. So there's no movement, there's no advection, no diffusion. It's just trapped there. So this is just a linear model for adsorption in porous media, and that's what we use also. And with these, we could simulate different mechanisms. So here's a normal heart, and this is the pressure. So this is like a slice of the left ventricle of a patient. What's the pressure gradient, how it looks like, the pressure map. And this is like towards obstruction, very severe obstruction. Here's like a small obstruction. You see here in this side, in this artery here, then you see there's you're gonna start to lose the gradient of pressure. And if you have a very strong obstruction, very severe obstruction here, you completely lose the gradient. So there's no perfusion anymore. So there will be no, so the tissue will really die here. And this is what we could simulate, and that's what we normally the medical doctors see, and that's what's I explained to you before. In the case of normal, the first pass, everything is very bright, and if you wait 10-20 minutes, everything is very dark, so there's no contrast anymore. If you have a problem of perfusion, there'll be some area that's dark, like before. So this will be dark because this region is not being perfused very well. But it's not severe, so after 10-20 minutes, still everything is dark. If you have a very infarct, so we have a very strong obstruction, So here the darkness increases. It's bright here, but it should be dark. Yeah, it's just like the contrast concentration is very low here. And then you see that the contrast is trapped here because this tissue is already dead. And that's what we could reproduce with our model. And here is the same model, but versus time. This is experiment versus, so the dotted ones are experimental measurements of contrast. And you see this oscillation because the contrast leaves the heart, goes again the heart. And we could also reproduce the oscillation with our model. And we could reproduce, like in the healthy heart, in the ischemic heart, how the concentration is like. And in the infarcted heart, also how the concentration, you see that the concentration of contrast after 300 seconds, 600 seconds, so 10 minutes, you have like a very high concentration because of contrast that shouldn't be there, but it's there because it's trapped by these, it's absorbed by the extracellular matrix, by the fibrotic region. So what we did next was to develop a hybrid model that is, because I mentioned that there's two levels of porous media and the interstitial space. The interstitial space is really small compared to the whole heart. Yeah. So the intercellular space is around micrometers. But the vessels, depending on the exam, you can capture. Like if you use computerized tomography, you can capture the main vessels. So we would like to use this information also. So we propose a model that has the vessels described as captured by computerized tomography, and the vessels then will, so the blood and the contrast will flow only inside the vessels that are described, and then we'll exchange, there will be some exchange of contrast to the extravascular domain, which is the interstitial space. So it's exactly the same model, just repeating here the same model, but the main difference here is that the contrast here, this equation here, omega I, is just the vessels. So it's not a porous media here anymore. Oh, sorry, this is wrong. So there's no phi here. So there's not a porous media here because we are solving this intravascular contrast in a given tree. So we have a given tree, and then we solve the intravascular contrast there. So this is the kind of experiments we did, and as I mentioned, we can extract these vessels from some exams, and then we can show here there is a block here, obstruction in this vessel here, and then there was this infarct, and the contrast accumulates here after a while, as it should be, as it's normally observed in these kind of perfusion exams. So next, what we are doing is with these new students. So this was a former student of mine that already finished, João Gabriel, already finished his PhD. So the next student, Evandro Gaio, is working with the same models, but looking at COVID-19 myocarditis disease. And because also there's a long, even during the acute phase and also during long COVID, you have a lot of reports of myocarditis disease. So you have inflammation in your heart because the virus enters the heart and start to damage. So the virus enters the heart, actually it starts to damage the heart because the immune system tries to kill the heart, the virus, but the virus are always inside the cells. So when it tries to kill the virus, it kills a lot of cells. So it's that's why it's very complex to manage drugs for virus. Antiviral drugs are very, very complex always, because the virus are always inside the cells. So if you want to kill the virus, you kill the cells also, like your body does. So it's a very tricky way to develop antiviral drugs, which is the case for COVID-19. We don't have yet an antiviral drug for COVID-19. Fortunately, we have vaccines very fast. After 1 year, we have good vaccines for it. But this is what Evandro is doing, based also on images of perfusion or treat. Now we also move to 3D. So we're also working with infarct and COVID-19 in 3D hearts. And this is the hybrid model. So we have vessels and the porous media, and then we can reproduce different diseases like myocarditis or the infarct one. So I don't know how many time, I still have 10 minutes or so, yeah?
Yeah, you have.
Okay, so then I go to the next part of the talk, which is edema formation, which is the first phase of the myocarditis. So before the cells start to die, what happens is that there's a lot of water accumulation in the heart. And this water accumulation is called edema. And this is also very bad for your heart. And it happens because of the immune response, the interaction between the virus and the immune system. So we coupled a model we had for immune system. Now we have published recently also, just a couple of weeks ago, a new model for COVID-19 response for immune system. And then we have this immune system inside the tissue of the heart, and the water also normally is evades the vessels, yeah, goes to this interstitial tissue and expands. Yeah, there's a water accumulation, expands this interstitial space, and we just call edema. And this is the process. So in the end, we have how to model this is we use classical formulation for deformable porous media like BO from BO formulation. And I'm going to show you here a previous example of this formulation, assuming linear deformation, which is not the case in the heart. And then, if I have time, I'm going to just show you another paper we are submitting with the model using hyperelasticity and large deformations, which is the case of the cardiac tissue. So this is the first image I showed before, like in the beginning of the presentation, a lady with COVID-19 and myocarditis. So this is another kind of exam. It's called T2 magnetic resonance exam that can quantify the amount of water. It shouldn't be there in the heart. So this is called edema. So there's a lot of water here inside the heart of this lady, and this can make a lot of complications. So we want to model now this phenomenon. It's a little bit more complex because it involves other cells also. So it involves the cells of the immune system. So what happens is that if you have pathogen, a bacteria or a virus here, so there'll be, what happens is that there are some macrophages here, or just called like leukocytes in general, yeah. So there will be some leukocytes here that will make an alert. Oh, I saw something wrong here, a bacteria or a virus, and will exudate some cytokines. And these cytokines will make the increase of the permeability of the vessels here. So the vessels have increase the permeability, so that neutrophils, another kind of leukocytes, can get outside of the vessels to the interstitial space. But so this is very, it's necessary. You have to bring more leukocytes to the place where there is this inflammation. So where there is virus or bacteria. But when you increase the permeability here, what happens is that there is water also going out. So you have you need the more leukocytes to come out, but then also waters come out as a side effect. And then there's if it's a big inflammation, so there will be a lot of water coming out. And this is what makes the edema formation. So we want to model all these effects here for using porous media. And then water will come out and like microscopically, it will deform your porous media because it will, it will like stretch these interstitial space. Yeah. So this is like what we had here, and we published 3 years ago also. We have like the virus, we have the immune response, the neutrophils coming to the virus. Then we have fluid accumulation after some time, and here you have the stress. So we have here the tissue deformation due to this accumulation of water. Again, one shortcut of the left ventricle. This is what I'm showing to you here. So we published this 2 years ago using linear elasticity. First, a little bit of what looks like the immune, the kind of models we have for the immune system. Here is for the colony of bacteria, and you see it like sometimes the colony can defend themselves. Neutrophils are in green here, and they they protect themselves. But sometimes the neutrophils manage to invade this colony and destroy the colony of the bacteria. So we have these, for a while we've been developing models that can do this. Here you have bacteria, and the bacteria is running away from the neutrophils. Sometimes the bacteria destroy the colonies. Sometimes the neutrophils cannot destroy the colonies of the bacteria, so it's a very complex system. And here's the fibrosis that protects the bacteria. So the bacteria are very, they have a smart way to defend themselves. They know that they produce a chemical fibrogen that is called that makes your own tissue to create that extracellular matrix that I mentioned. That's called fibrosis. Yeah. So they generate fibrogen so to protect themselves, to create the barrier. But who creates the barrier? It's our system. So it's a very smart way to protect themselves. Sometimes they can protect themselves, like here. They manage to make a very good barrier, and then they still here. They have a colony. The neutrophils cannot destroy it, but sometimes they don't have enough time to make these. Oh, maybe I should. So sometimes they do not have time enough to make these barriers. Neutrophils are always coming after the bacteria and trying to destroy it. And sometimes the neutrophils manage to destroy the bacteria before they make the barriers. Sometimes they cannot destroy the bacteria. So we have these kind of very nice models for the immune response. And we couple these kind of models with porous media, with deformable porous media. So this is the bacteria. Now we have in a porous media, phi also varies with time, is the fluid phase here. And we basically have the bacteria only diffusing, it's moving randomly. Yeah, so random move, or it's normally pictured by diffusion. You treat the random walk, yeah, like Brownian walk with diffusion. That's how we model. But the neutrophils are smarter. So the neutrophils, they they really go after the bacteria. So they're always chasing the bacteria to try to kill them. So a way to, so they diffuse also. So there's some random walk of neutrophils or leukocytes, but there is a directed, so there's an advection. And advection of neutrophils is in the direction of the gradient of the concentration of bacteria. So this is called chemotaxis. It's just an advection of bacteria, but it's the neutrophils goes towards the bacteria, and the way to model it, it's with advection term of velocity proportional to the gradient of the concentration of bacteria. So it's a very common way to model a phenomenon that's called chemotaxis. And that's all, that's boundary conditions and so on. And here's the very simple deformable porous-media BO equations for that. So you have like deformation, so displacement, you have pressure, and again here you have the here we make an assumption that everything is stationary. Darcy law, it's not stationary, sorry, but you have the movement. It's Darcy law, so the velocity of water also is captured by Darcy's law, and his this would be like the mass equation for water, and here the moment equation. Yeah, so we have these two equations: balance of moment, balance of mass for water or for the porous medium. And here we have like we use, it's amazing that there are these very old models already for fluid exchange in capillaries, like for Starling law from 1896. That's what we use. So so water leaves the vessels depending on pressure in the capillaries and the pressure in the interstitial, and also depending on these pi C and pi I are like reflection, like proteins inside and outside that makes osmosis, or how to capture, how to model osmosis. Yeah, so Starling law is here is mainly modeling osmosis together with a linear exchange of fluids because of the difference of pressure. That's a Starling's law. That's what we use. We use like the hydraulic permeability, as I mentioned, will increase when you have virus or bacteria. So that's the way we increase the permeability of the vessels here using the Starling law, and this is coupled here. And also the reflection coefficient here, sigma, also depends on the bacteria inversionally. And we could also, again, we developed a patient-specific model. So this is edema formation from a specific patient. Now it's a long axis. Here's the apex of the ventricle. Here's the base of the ventricle. I was showing before a lot of short axis. This is a long axis of the heart. And here we generated the mesh, we generated that. So this is like what was done by two medical doctors. They identified manually, like this is the edema, and this was using the image and using our model. So all the process of, so we started with a single point here of bacteria or virus, and let the system develop, and this is what we got as edema formed after stationary. It goes to stationary. So it's very similar to what was captured in the patient. So it was very nice, surprising. It's always a very good surprise when everything matches with such a simple models. Yeah. And now we are, as I mentioned to you before, now we are going to using a better formulation for large strain for elasticity. together with Ricardo Bayer from Australia, and we are working together with him, developing this kind of more complex and models, because tissue is not that hypothesis that cardiac tissue was linear, assuming deformation as linear elasticity is not a very good. Cardiac tissue is not, there's a large deformation and not linear at all, the response. So here we are using more complex models for this, that are more appropriate to model the strain stress relationship in cardiac tissue. And here is the 3D. You have this is the wave of pathogens. Here, in this case now, here's the virus already, and here you have the virus, and it's being defeated by the immune system. It always goes around, managed to move fast around, and so in the end, since it moved around all the heart, water came all over the heart. So it's something we managed to reproduce. It's also present in the clinics. You have localized edemas sometimes. So there's only edema in one point of the heart. It's localized. Sometimes all your heart is edema. So there's water all over your heart. And this is, we could explain how we could get water all over your heart, because the pathogen managed to escape the leukocytes and whatever. When the leukocyte is entering, there's water entering. So there will be water entering all over your heart. And so you have it's called diffuse edema, the clinicians call it, or I just call it global edema instead of localized edema, but the medical term is diffuse edema. And this is what we just finished and we're about to submit. So that's what I had to show you today. Thank you very much for your attention.
Thank you very much, Rodrigo. Thank you.
Thank you.